Okay, so this time I'll go into my formal presentation here on epidemic viruses in nature and in silico. And the message here is, and I think the message of the whole day is, what we're trying to do is, to the best of our ability, represent what's going on in nature in computational representations, models or simulations, to help us think about complicated things. I view models as supports, as ways to help us think about complicated things that are otherwise if we were reliant on our own brains would just be too damn hard to think about. So I'll start with a bit about viruses as biological entities, the origins and spread of HIV. I've done quite a bit of work on this myself, and so I thought I'd give you sort of the first person in the field look at this. It's not computational, but it's the biology. A bit about the origins and spread of flu, viruses as computational entities, and then I'll finish with a few philosophical reflections. For those of you who aren't virologists, there are a number of families of viruses that are shown here. This is their morphology and biochemistry definitions, is that they have names like orthomyxoviruses and rhabdoviridae and things like that. But we tend to know these by their common names. We'll start to sit down here. Of influenza or Nipah or hantavirus or SARS or poliovirus. But the ones that we're going to talk about today are influenza and HIV/AIDS. And what these viruses all have in common is that they have an origin in animal species. They have made a jump from animal species into humans, and they can evolve rapidly in humans and can transmit from person to person. The simple idea of global emergence of epidemics is that there's some animal reservoir on the bottom. There's a species barrier. The viruses jump across the species barrier, and then there's gradual spread across the entire population. And of course, one of the objectives is to try to keep this from happening, to keep it at the lowest level as possible, to stop the epidemic from ever beginning in the first place. Let's start with HIV/AIDS. I had dinner last night, and I told people that I worked with Jonas Salk on AIDS vaccines for a little bit, and so they said I got to see the pictures. So I'm putting it in here. Just Jonas Salk developed the polio vaccine at Pittsburgh, and so I'm the Jonas Salk Professor of Global Health, and it was a privilege to work with him a bit on this. This isn't about Jonas, but I had to put the picture in. That was when I was on active duty in the U.S. Army. I was at the Walter Reed Army Institute of Research for many years, where I worked on vaccine development. The global AIDS pandemic is now going past its 30th year of recognition, with probably pushing somewhere on the order of magnitude 100 million people infected worldwide. And I have to say, of all of the disappointments in my own professional career, this happened on my watch. I'm an infectious disease physician by training, and somehow I and my generation let an epidemic go on with 100 million people. And I think that's unacceptable, and I never want to see that happen again. So part of my motivation here is understanding these processes so that it doesn't happen again, so that we are smarter about it the next time around and intervene before we reach 100 million people. We just can't have that kind of thing happen. And I worry about this. Has it happened once? It could happen again. Whether it is going to be a SARS-like thing, which is worse, or an Ebola-like thing, which is worse, these are not hypotheticals. They happen. And so we do need to think about them. If you look at the different type of HIV-1s, the genetic variants of HIV-1, by genome sequencing, you can look at homologies, and you can put them into clusters or clades, as they're called. And so I did a lot of this early on when I was at Walter Reed. We were looking at the epidemic worldwide. And whenever there was a new outbreak anywhere, I went there personally and collected viruses, brought them back. We sequenced them. So in 1986, I went and got the first C genotype viruses out of Zambia, and then we showed that was a new genetic variant coming there. And then in 1989, the same thing in Thailand. And I'll spend a little bit of time on the Thailand. But the point is that each of these epidemics in different locations has some different genetic variants of viruses. It's not just a single epidemic. There is this clustering. And in some ways, it sort of was the same kind of fragmentation that you were talking about in the game, is that not everything is randomly mixed, that an epidemic is coming here, an epidemic is here, and these tend to be confined within a geography and social nets. In 1989, it started having, there were first detection of HIV in Southeast Asia among military recruits at the Gavila Army facility there. I had lived in Bangkok for 6 years. I know Thailand pretty well. I speak Thai reasonably well. So it's sort of a second home for me. So they asked me to come and see what was going on. We obtained viruses from these recruits and isolated a new virus. Within 2 years, 15% of all the young men in northern Thailand were infected. It was just explosive. It was, we watched that and we said to ourselves, my God, the country is going to be completely decimated, so is all of Southeast Asia in a matter of a few years. Fortunately, it slowed down, and fortunately, Thailand did an absolutely terrific job of containing the epidemic. But when we looked at the viruses, and these are the sort of genome maps of the different clades of HIV-1, the major groupings are the clades A, B, C, D, and E, and I've color-coded them to make it easier to. They don't come in these colors really. And then if you look at and so that's just the genomic organization at the top, and then on the bottom is a diagrammatic representation of the consequences of that. And what you see is on the lower, at about 2 o'clock down on the below, is the clade E, in fact, is a recombinant virus where half of the virus was from the A type, and then there was some sort of recombination event, so a new envelope got swapped onto the surface of the virion. And that implied a very important observation that the virus was recombining as it was evolving. It was swapping genes and evolving. It was trying out new strategies to spread. And subsequently, there have been lots of recombinations shown worldwide. Well, we found this E genotype in Southeast Asia and the C genotype in South Africa. And then when we went to Central Africa, we found all of the viruses there. We had a collection that was from the Congo Basin, and we found all of the genotypes and recombinants of these types. And so the question is, where did these viruses come from? If it's all coming out of equatorial Africa, what was the origin there? And so I became friends with the head of the National AIDS Control Program in Cameroon. I hadn't been to Cameroon, but he was a good guy, and he invited me, and so we ended up doing some work. He's the, let's see if I can show the, I can't, about there, the heavyset guy with the black baseball cap in the back. His name was Colonel Mputi, terrific guy. Just got a good sense of humor, but really cared deeply about what was going on in his country. And so we worked together and went around and set up studies in Cameroon, in the jungle, looking for what was the origin of the AIDS virus. Standing next to me in the yellow hat is Ndongo Toromiro, who was a postdoc with me, and she's subsequently gone back to Cameroon doing very well. Trying to do studies in, if you've ever been in the jungle, is not easy. It looks romantic when you look at the sequences, and it looks so clean and so neat. But this is what goes into that stuff. This is the real world. Now, of course, you have to ask, who's taking the camera, and where is Don Burke? Who's taking the picture? And yes, I'm taking the picture. I'm not doing the. But nonetheless, this is what it's like to get out into these areas. And when you go to the villages, you have to work with the people there for, it was a matter of weeks before we would actually do studies. And so we would have festivals and dancing and things like that. And then we would finally go into full informed consent. It was quite an undertaking. But the idea was looking for this animal reservoir. What was the ultimate origins? And if you've ever been to equatorial Africa, that people eat animals in the jungle. That is their source of meat. There is no other source of meat. There's really no animal husbandry to speak of, and there's no commercial sources of meat protein. And so if you just drive along the road, I was in one village and asked, show me what you caught today in the village. And you see there are these, starting on the left side, there's armadillo-like things and antelope-like things, but there are 3 monkeys there. And people routinely eat monkeys and other sorts of non-human primates. In fact, it's a major cause a major source of meat protein for people that live in the jungle. Most of these people are very poor. They've never heard of the idea of endangered species. You know, if you talk to them about endangered species, they've literally never heard of the notion, let alone thinking they may be doing something illegal. And then you see the guy on the right, he was the local school teacher. I asked him, show me how you prepare the material. The monkeys, you see that long skinny thing, that's the monkey's tail. And what he's chopping on with his machete there is what's left of the carcass of the, but he's got his bare hands there as a machete, there's blood everywhere, and so if the animal's infected, it's not, you don't need much of an imagination to figure out how he could have been infected with the virus from the animal. And if you then do get viruses, so you go looking for viruses in these, first you test for antibodies, and then you isolate viruses, then you sequence viruses. And here's a comparative phylogeny of viruses from the polymerase genes of viruses from chimpanzees and from humans. And when you do that, the viruses of the human HIV are fairly similar to the virus from the chimpanzees. And so that's the first hint, that there was something going on. But that still doesn't tell you, how does this occur? Well, the bushmeat hunting looked like a pretty good possible route. And so we wanted to see, do in fact people who are hunting, butchering animals become infected in that process? And so there's a whole lot that went behind this, but here are a whole lot of genetic sequences from the genomes of viruses that were infecting animals, and the species are along the right-hand side, the names of different types of non-human primates. But then there are 3 boxes there called CAM viruses that we got from people who were hunters. That we isolated, and they were the viruses that were virtually identical to some of the viruses that were in the animal species. And these were people who were hunting those animals. So it was pretty good inferential evidence that persons had become infected in the process of hunting and killing animals. And in fact, it wasn't that hard for us to find them. These weren't AIDS viruses. These were retroviruses similar to AIDS, which said that the mechanism of chronic virus in an animal with butchering could cause cross-species jumps. And here are some of the animals that where we found people infected with viruses from these animals because they had hunted and killed and butchered them. And in fact, when we did some serologies based on how many people had antibodies against some of these animal viruses, our best guess that in the people that are forest-dwelling people in Central Africa, about 1% are infected with zoonotic retroviruses, meaning there's an awful lot of people out there who are living in the jungle who are getting infected routinely in the process of their daily living, which is, this is what you have to do to eat. And therefore, most of these cause no disease as far as we know. Most of these are not transmissible from human to human as far as we know. But they're one step away from adaptation, and these are sort of the next emerging viruses. These are the ones that you want to watch and to say, if we're going to predict where the next one comes from, these are the ones I want to know what's happening. And so it happens regularly, and it's probably due to hunting and butchering. But one of the lessons we learned is these are not rare events. Viruses are jumping all the time in the Congo Basin. People that hunt animals get infected with those animal viruses, and they jump easily, particularly from non-human primates. So we didn't study all of the other viruses. We didn't study the viruses of armadillos or antelopes to see how frequently they jumped as well. But that's something I wish we could do. But our hypothesis was that it was more likely viruses adapted to other primate species would be able to jump to human primates as well. And we call this viral chatter just because it's like terrorism chatter. When you find viruses jumping, you get nervous about it. You don't know exactly what's going on, but you want to pay attention. So let's turn our attention to the same issues with influenza. Influenza is a virus that has a structure with envelope proteins, neuraminidase, and 8 genome segments in its that encode for its viral proteins. This is another virus that can recombine. If you have a single cell, again, I've colored the viruses here of flu, red and green. They don't come in those colors. And if they co-infect a cell, then there can be reassortment of the genomes, and you end up with these hybrid varieties where it's a human influenza virus with an avian. And this has happened now with the viruses that are H1s, and the concerns are that you'll get reassortments between the H5s and the H7s and all the other avian flus that are jumping into humans. So you have these viruses jumping into humans, but they haven't reassorted with the human-adapted viruses yet, and they could any day. So one of these days, this could happen, where you get a reassortment, which is a major, where most of the virus is well adapted to the human, and all it's done is changes its surface envelope protein and has this adaptation to be able to spread. It's been known for a long time. This is a cartoon in Ed Kilbourne's 1958 textbook about the different types of influenza viruses in different species. Almost all of the species, there's a wide variety of influenza viruses in avian species, H1 through 15, and then some of these are common to both humans and animals, H1, H2, H3. And then H7 has been found in horses. And so there are, and any one of these things can move back and forth. Influenza has this ability to jump across species and potentially cause epidemics, mostly in avian species. But there's a huge reservoir in the animals, just in some ways waiting to jump, and we need to be prepared for when the jump does occur. These are the ones that we've seen in humans in various types of outbreaks. Particularly the H5 and H7 have caused a lot of concern in Southeast Asia. And we need to be prepared in case those do turn into significant epidemic viruses. But again, the same idea here is that there are lots of jumps going on all the time, but most of them don't go anywhere. Most of them don't adapt. Most of them are not transmissible human to human, but they could. And so an important part of understanding what's going on is the process of adaptation. So there is one underlying message behind this. The viruses that tend to do well and cause epidemics are evolvable. And they can evolve, they can swap genes. They're almost evolution machines. They're like they've got a little artificial intelligence of their own. Is that they figure out how to adapt, and then they swap genes with another virus that has also partially adapted. And if you look at the process of emergence, we need to be smarter about figuring out how we can at every level intervene in the emergence of epidemics. So starting at the bottom, reducing infection in the reservoir animal species. It's not going to be easy. I don't know how we're going to reduce infections in gorillas in Central Africa. That's going to be a challenge. But we can reduce infections in avians, in chickens and ducks and things like that. The Chinese have been using vaccines in a lot of countries in Southeast Asia, H5 vaccines for some time now, trying to reduce H5 influenza. We can try to prevent cross-species infections by being smarter about how we deal and make sure there isn't blood-to-blood contact or other direct contact with infected species. We can try to prevent adaptation to humans if we can find out people who are infected early and do something about it. I use this term quenching. If you've got a local outbreak, can you stop it before it begins? And we'll spend more time today talking about epidemics that have already emerged and how you can try to contain them, which is, I think, where most of the effort is now. But I've tried to push this notion of pushing the whole idea of prediction and prevention as sort of the next place we should be going in terms of epidemic viruses. Okay. Well, this is one way you can prevent cross-species infections, just have absolutely no contact whatsoever. He loves the petting zoo, of course, and he's only allowed to touch the animals through a containment kind of. And here's another approach to stopping transmission. This is the students who I was with at dinner last night. I told them about this, and so they made me do this. I want you to know. So let's give it a try and see how it goes. We all sit in the back.
The Public Health Agency of Canada proudly presents coughing, sneezing, and you. Every time a person coughs or sneezes, millions of disease-causing germs are launched into the air. Polite people cover their mouths, but using your hands can spread germs to other people. Until recently, the best advice was to use the inside of your elbow. Germs get trapped in the fabric and die. But we now know that even these precautionary measures aren't always sufficient to prevent contamination. That's why your best bet is to sneeze into the back of your knee. Here, people are far less likely to come into contact with your germs. Can't reach? Then use the back of someone else's knee. They'll be glad you did. By following these steps, we can all help make the world a healthier place to live. This has been a presentation of the public.
There's the video's online. I think if you want to, I should give you a website for it, but it is a lot of fun. But what it does is it, in some ways, jokingly talks about what do we really know about contact and what is the form of contact and how much is due to hands and how much is, and I have to say that's still a very rough art. Is that I personally am pretty, frankly, skeptical about the role of Purell, hand sanitizers in influenza transmission. I really think there's very little evidence for that. We've done one major study in schools, and we had a hard time proving efficacy we showed marginal efficacy for some people, but not for most. So that's an important topic. So this notion, even though it's a joke about what's the right route of transmission, I hope I made the point. So computer simulations can help us think through these hard problems. So how do you make decisions on the far right there? How do you make a public health decision that's a wise choice? Well, if you're a policymaker sitting in a state capitol or in a local health department or in Washington, D.C., how do you decide? Well, you use your own prior knowledge, you use expert advice. One of the ways to make policies, you sit around and you say, I don't know, George, what do you think? And he says, Don, I don't know, what do you think? And you make policy that way. But I think there are better ways these days, particularly when you're dealing with epidemic dynamics. What you can do is you can take the data, and instead of using your mind to analyze it, you make a computational representation of that, and then use that to evaluate policies. It's an alternative way, in some ways, that computational models can compensate for weaknesses in mental models. By the way, every decision is based on a model. Everything you do is a representation, but it's a mental model. And it's a mental model that is in some ways inexact. What goes into your mental model, you really don't know. You really don't have your parameters. You really are not explicit about what goes into your mental model. And a computational model forces you to be explicit. It forces you to understand what are your assumptions, what are your parameters, what do you really know, and what you really don't know. So I'm a big believer in the idea of using computer models not as to make the decision, but as an aid to your own internal mental model decision making. So you can do all sorts of cool things, and you'll see more of this. So we did a lot of this over the years of representing all 300 million people in the United States as individual entities in large-scale agent-based models, put them into interacting societies of where people move, how they go about their daily business, and then do the sort of this artificial injection of a flu or a transmissible agent into the population, and then see how it spreads. And then what we do once we've done that is then we'll say, what would happen if you would close the schools, or what would happen if you would stop the airplane travel, or what would happen if you vaccinated everybody over 40? And you can do that kind of thing. You can go through these thought exercises explicitly in computational representations to help you make decisions. So it's a pretty powerful technique. I personally like to see maps and red spreading stains and things like that, just because people think that way. That's the way, that's my mental models already, and so the computational model maps onto my mental models. And that's the way most policymakers think too. There's a whole separate science of how do you communicate computational results to decision makers, and how do you translate it in ways that it is acceptable as a help to their mental models as well as to your own mental models. So epidemiology in silicon has a number of practical applications. The main one is as an interactive simulation to evaluate and inform policy decisions. Another is a pedagogical device to train public health students. It helps you think and understand what's going on. Another is as sort of an experimental epidemiology to construct and test out field trial designs and things that are for complicated interventions. And it's an analytic tool to improve understanding about real world epidemiology. There are a lot of reasons you would model and do simulation, all of which are important and valuable. So let me close with some philosophical reflections about modeling. This is one of my favorite paintings by René Magritte, sort of a painter-philosopher, and the caption reads, "Ceci n'est pas une pipe," in French means this is not a pipe, and of course this is not a pipe. This is a painting of a pipe. It's a representation of a pipe, but he says, he writes it there just to point out that this is a mental model. This is a way to talk about pipes. This is how you and I communicate about pipes. This is how we parameterize pipes. This is how we represent pipes. And so I just want to have you keep in mind, ceci n'est pas une épidémie. This is not an epidemic. I know that. You know that. This is a way of representing epidemics. It's a way of talking about it. And it really is in some ways the same notion. How do we just communicate? It's a device for representing ideas to each other. And this one happens to be dynamic, happens to be computational. But it's the same idea as René Magritte, is that we need ways of expressing ideas commonly. So with that, today's agenda, we will be going over my talk first, and then we'll spend time on HIV/AIDS with both aspects of behavior and computational network analysis, and then we'll be doing sort of the same thing for influenza, and then we'll finish with a panel discussion for the day. So I hope I've set the stage for where viruses come from and thinking about computational models. And if nothing else, you'll never forget that film. So so I think we have a little bit of time, do we?
No questions.
Yeah, okay, good. So please go ahead.
Are there any mathematical models for HIV/2, or is HIV/2 not that big?
Yeah.
Hello. I just wanted to know if there are any mathematical models for HIV/2, or if HIV/2 is just not that big of a concern.
Yeah, I'm not personally aware. George, do you know anybody who's modeling HIV/2? Doesn't mean you couldn't do it, and it largely is that by comparison it's dwarfed by HIV-1 in terms of its epidemic importance. It has some different epidemiological properties than HIV-1. It has a different origin. It has a different species that was the cause of it. And as a consequence, it isn't quite as transmissible. It doesn't have quite the disease manifestations. And the total number of persons worldwide is substantially lower. So, but if somebody wanted to, if you wanted to do an HIV/2 model, you could do one. It's just that no one has invested the energy.
I had a question. I was on Facebook, and one of my cousins had sent me a link saying that they were thinking about bee venom killing the HIV virus. I was wondering if you knew anything about that.
Let's see, where do I begin on that? One of the problems with something as terrible as the AIDS epidemic is there are lots of people who step forward with what they say are cures or treatments or new ways to stop the epidemic, particularly before there were real antiviral drugs that work. There are drugs now that really do work, and it really is possible to keep somebody in a good state of health for a long period of time. But I can't tell you the number from between 1985 and 1993 or thereabout of claims for whether it's bee venom or things like that, most of which are well-intentioned. People think that they see it as causing an effect in one person who happened to have an unusual course. But for the most part, they don't pan out. So I would have a level of skepticism that would be very high for that. Now, is it possible there's some component of that, that maybe that we could isolate and then use to study? Sure, I would think about that. But the sort of the naturopathic ways of treating HIV so far have not been not impressive. George, you want to comment on any different than what I said?
No, in some ways, the whole unfortunate history of President Mbeki not accepting the HIV causal hypothesis resulted in a lot of homeopathic approaches in South Africa and really delayed the rollout of antivirals for a good 5 to 7 year period, and really was very unhelpful in terms of getting the epidemic under control in South Africa. So, there's a lot of focus on alternative approaches, which in some cases may be very complementary to standard treatment. But when we know standard treatment is so effective, if you just rely on those complementary alternative approaches, you could end up in serious trouble. And the Mbeki example is probably the worst case example in terms of delaying effective interventions and then resulting in a much worse epidemic than if they had aggressively treated early on.
I can come up with at least 2 other good examples of where they were egregious. The Buddha and Mubarak treatment and the Moi treatment, and there are a whole bunch of others that are. So, but we keep looking, and maybe there's something to that. I don't want to be too skeptical, but I would think I was pretty damn skeptical.
I was curious about the bushmeat sort of thing. That was a lot of the old world primates sort of thing.
Yeah, right.
Is there anything with like the new world primates?
Yeah.
Because that's definitely not an exclusive
Sure.
bushmeat trade.
Yeah. So there are none of the lentiviruses are naturally found in the New World primates as far as we know, but there's some of the other retroviruses are. So that particularly there are the HTLV1, 2 class, the PTLVs they're called, are found in the New World. I don't know anybody has shown evidence of humans becoming infected with those viruses. In Central Africa, we've found several hunters who've been infected with viruses from those other animals. But the basic point is whatever's in the animal species, if you hunt it and kill it and you get blood on you, there's a chance that you're going to get infected, and we need to understand what are those processes. So my former student, Nathan Wolf, he became sort of famous for doing this. He set up a network worldwide, not only in Africa, but in Asia and South America and in several other countries. And to do exactly that kind of thing is to look at indigenous peoples who are dependent particularly on hunting, and then to try to understand what are the potential reservoirs. So it's a general principle. In the back there.
So what is the physiological effect on the primates, and can you get any insight into treatment for humans from the actual primates that are infected in the wild?
Yeah. So particularly in the viruses that are like HIV, the SIVs, in a number of species, they are fairly similar, and they can cause AIDS, particularly if they're not in the native species. If they are given, if you take a virus from a sooty mangabe and put it into a rhesus monkey or something like that, it can cause disease. And so there are lots of people around the world who are using the SIV models to investigate the pathogenesis and to understand ways of intervening. And it's turned out to be a very, very productive way of understanding immunology and pathogenesis of HIV by using the SIV systems.
With the epidemic in Thailand that you were talking about with military recruits, how exactly did they quarantine? You said that they took care of it pretty quickly. How did they do that?
Yeah, so Thailand, as a country, adopted a 100% condom promotion campaign across the entire country. Although it's not published, and I guess this is being recorded, a majority of the first 13 young men that we got the E genotype virus out of had all been to a single massage parlor in just north of Chiang Mai. And if you're talking about patient zeros, there can be patient zeros at every level. So what we thought was there was probably somebody who came from Central Africa to the Chiang Mai area with the E genotype virus, that he probably went to this one particular massage parlor, had sex with a worker at that, and then It was subsequently transmitted to the point where it exploded. There really was probably a patient zero for the E genotype in Southeast Asia. There was a patient zero, if you've seen the movie, the band played on, and this idea of the patient zero of HIV in the United States that was thought to be a person who was an airline steward and transmitted a lot of the B genotypes around the United States. So each one of these outbreaks was a separate clonal outbreak. In Thailand, as I said, they were very effective in dealing with the epidemic by essentially promoting safe sex at a national level and with an astonishing effectiveness. I think it's fair to say that it's probably, and George, I don't know if you'd agree with me, probably the most effective national level campaign anywhere in the world. And that's a really interesting discussion about why was Thailand successful and other countries have not been successful. And that's we can talk about that offline if you'd like to. You agree with the analysis, or okay? Okay, one more out there, and then we can finish.
Most of the time when they talk about medications for HIV, it seems they're dealing with vaccines, right?
Yeah.
And they're looking at getting a vaccine to prevent
Yeah.
people from getting infected.
Right.
Now, when you give an retroviral drug to an individual that is infected, it probably lowers the viral load, which
Right.
makes the what is it called, the infectivity
Yeah,
lower.
right.
So they're less able to transmit it. But have they ever looked at creating something for the person that's infected that doesn't lower the viral load but can lock up the HIV from not transmitting it to someone else, instead of giving a vaccine to the person and then saying, okay, this person is gonna give it to you, but can your body protect it with this vaccine? But
Yeah.
give a vaccine to the person who has it to just stop it from jumping to another person.
This is not a setup, but I have written several articles and book chapters on vaccine therapy for persons who are infected. The idea has been around for a long time of the idea that we could somehow boost the immune system so that it could control the infection by doing exactly what you're saying, is that by providing a vaccine that somehow alters the host immune response in a way. And people are actively working on that, have been for some time. We did trials with the very first genetically engineered envelope proteins. That was one of our first thoughts. This was at a time before there were drugs, and so it was our only alternative, was to try to use that approach. We were able to alter the host immune system, but it didn't have any lasting effect on the ability to control the infection and didn't have any effect on the clinical history. I think it's still a good idea, and I'm glad that people are still pursuing it, but it still hasn't really been shown as an effective. But the idea that vaccine therapy is a potential adjunct to chemotherapy is still a viable scientific idea.